Over closer to the door. There you go. Let's move the table closer to the door so it's easier for people to get to. A couple of items that may be of interest. These are supplementary, not required, just if you'd like to look at them. I may have mentioned when we were doing integration and metabolism that the tables of glycemic load and this exploration about do we even need carbohydrate at all, which are kind of interesting in thinking about the integration of metabolism. But more recently, I want to draw your attention to two items about ovarian cancer. A member of this class drew my attention to the fact that just as we were finishing up the connection between genetic predispositions and cancer, that there had been a report on identifying genes that significantly contribute to the probability of cancer for ovarian cancer specifically. And what I've done here is included a link. Why don't you take a stack? Take some with you and pass them. The CNN report of it and a slightly more detailed one that goes a little further into the genetics behind it. So a very interesting coincidence that we should have synchronously the report of one of the more scary cancers that women can face and the genetic factors behind it. Are there any questions before we go on about the DNA repair and cancer chapter? Okay. We will then get rid of this file browser and start up. Here we go. Okay. So we've discussed DNA replication. We've discussed DNA repair. DNA by itself, I once got Ben Lewin very mad by saying that DNA is the dead hand of the past. DNA by itself does nothing. So we need to complete our macromolecular metabolism part by thinking about the rest of the big molecules. And today and Monday we'll be discussing the material in Chapter 19, which is transcription and RNA processing of various kinds. How you get information out of the gene and start getting it into a form that'll do some good. On April 29th and May 1st, we'll cover Chapter 20, which is on protein synthesis, protein processing. And these are the workhorses. These are the macromolecules that do 90% of the work in the cell. We'll be set up then on May 4th for the last hour exam, and it'll cover the last 4 chapters that we've discussed. May 6th and 8th, I will be giving overview and integration sessions covering the whole of the course to try to give you kind of a synopsis, a refresher of the material we've covered, the ways they tie together, and so on. And then the final exam will be May 11th on Monday at 2:15. Again, I hope you're beginning to review for the final already. It's probably not realistic to try to do a comprehensive final review in just 2 or 3 days before the final. So, any questions about the mechanics of the course at the moment? Okay, let's do something interesting like science. Your textbook introduces this topic with a mushroom. And this mushroom has the interesting name Amanita phalloides. And its other name is the death cap mushroom. And the reason they choose to start with this mushroom is that this mushroom produces a antibiotic called alpha-amanitin that has been a very powerful tool in understanding how RNA is synthesized, because it's a peptide antibiotic that can inhibit RNA polymerase, the enzyme that makes RNA. And it has specific ones that it inhibits. So if you inhibit RNA synthesis, the RNA you have starts to decay and be degraded. As it goes away, protein synthesis gets stopped and the proteins eventually go away. Interestingly, alpha-amanitin makes 2 terrible, poisonous, horrible things. It makes alpha-amanitin, which inhibits RNA synthesis. It also makes phalloidin, so Amanita phalloides. It's the other toxic thing. And phalloidin polymerizes your actin so that someone who's eaten the death cap mushroom has the actin in their liver polymerize, and the cells of the liver look like little urchins, and they pierce themselves and you dissolve your liver. So it's not very good. You actually die from the actin antitoxin, not from the RNA inhibitor. Little side note on how important it is to keep in mind the properties of these things. These mushrooms actually taste very good, apparently. A friend of mine who gathers mushrooms was driving in Rhode Island, and he saw an old man by the side of the road picking what he could see from the car was alpha-amanitin, or Amanita phalloides, rather. So he felt morally bound to stop and talk to the man. And he said, have you ever picked these before? He said, oh, yes. Picked them a lot of times. You do? What do you do with them? Oh, I eat them. They're very tasty. And my friend said, well, how do you prepare them? Oh, he said, oh, you have to know how to prepare them. You slice them thin, you soak them in water, and the water will turn black, so you throw that away. And you keep soaking water till the water runs clean, and then you pat them dry, fry them in butter. They're delicious. So evidently both alpha-amanitin and phalloidin are completely water-soluble. I'm not going to do the experiment, but if you're ever desperate and need to eat them, there you are. So transcription. Transcription is our name for copying over. That's what it means literally, is writing over, copying over. And what it does is transcription copies the information that is in the linear sequence of DNA into a linear sequence of RNA, and the RNA is the more active component of this system. The DNA by itself does very little. It's extremely important for transcription to be selective. If all the genes are transcribed at the same level all the time, it would almost certainly produce a chaotic situation in which the cell would have no way to cope with changes and so on. And so there's regulation involved. There has to be regulation to have an effective use of the genome. I'd like to try to introduce one of these places where the words of biology are a little bit hard to put your finger on. And I want to point out that a transcript is not exactly the same as a gene product or a gene. Transcript simply means a piece of RNA that's been made from a DNA template. Now, the kind of transcripts that we easily think of as gene products are the messenger RNAs that code for proteins. And messenger RNAs are going to code the primary sequence of the proteins internally. There are other transcripts that are genes without proteins. So any of the functional RNAs, a ribosomal RNA, a transfer RNA, a small nuclear RNA, sRNA in the ER, things like that, are transcripts with phenotypes, so they can be genes, but they're not translated. They don't have any protein product. Just to make life a little more complicated, most of these transcripts are only going to be functional if they've been processed in some way. So it's not the nascent, just completed transcript generally that's functional, it's the processed one. There are regions of DNA that can control transcription and therefore have a phenotype. And yet, and by having a phenotype, they are behaving as a gene. The original definition of a gene was a heritable unit of transmission that could cause a phenotype. And so if you have a region of DNA that can control other things, it's going to have an expression, a phenotype, even if it's not making a protein or an RNA. And then many transcripts have no effect that we know of. When we look at the transcription in a typical human nucleus, probably 3/4 of those transcripts never leave the nucleus, and at the moment, all we know is that they're being there. They may in fact not have any individual function at all. It may simply be a need to keep up a certain level of transcription to keep the nucleus inflated to a certain size. We don't really know. So it's one of those areas where there isn't a simple one-to-one correspondence. And I'd like to make it easier for you if I could, but life is messy in this respect. If we think about the human genome, it turns out that the human genome contains somewhere between 20 and 25,000 genes. And the number is shrinking. It was 30 to 35,000 as recently as 4 or 5 years ago. Before we sequenced the human genome, people thought we'd have 80 to 100,000 genes. So it turns out it takes fewer genes to make a human being than we initially thought. If you take that entire stretch of DNA, that meter-long piece of DNA, only 1% of the DNA of the genome has genes. The other 99% is other stuff. Sorry, I keep clicking. Most of these genes have introns in them. That is, they have sequence that's transcribed into RNA but then taken out for the final product. And we'll talk more about that in a little bit. Some of this tally of 20 to 25,000 genes are identified as open reading frames. That is, these are things that have all the features of a gene. They have what looks like a start site, and they have a translatable interior, and they have what looks like a stop site. So they are open reading frames, but we don't necessarily know what they do. Some of the genome is taken up with what are called pseudogenes. These are gene sequences that are clearly related to existing functional genes, but that have mutated into a useless form. They might have lost their start site. They might have they might be truncated. They might have an insertion that scrambles it with something else or whatever. But it was definitely a surprise that there were so many fewer genes than expected. So the absolute size of the genome, how much DNA an organism has, does not seem to be the best predictor of how complex an organism is. And probably a better number is the number of regulatory genes. That the more complex an organism is, the more likely it is to have genes to coordinate the behavior of all those things. This is a small segment of one of the human chromosomes. This is from chromosome 20. And what they've done here is to map for you some of the consequences of the Human Genome Project. So you can see here in the sort of purple color genes that were known in advance, and now they know where they are in the genome. The blue ones are novel genes that were not known before the sequencing process. The little orange arrowheads are pseudogenes, things that were genes once but aren't anymore. And a few green things here are perhaps genes, but we're still in the process of checking them out. You notice there's a lot of empty space between them. When we were talking about replication, we've made the note that replication isn't just sort of pea soup, but it happens in structured local areas of the nucleus. And when we look at transcription, we find that that's true as well. So this is a fluorescence micrograph showing you that replication, making new DNA, is happening in very concentrated, multiple little factories of DNA polymerase. And transcription is also occurring in a rather structured punctate pattern. And because we have different colors, we can compare these two, and you can see that they, in fact, are not in the same place. So that the nucleus has subregions within it that are specialized for these different functions. And if we look at DNA polymerase RNA polymerase schematically, this is a nuisance. Maybe if I put it on the counter. Let's try that. All right. If we look at the RNA polymerase, obviously it's going to have to be able to interact with the DNA. It's going to have to separate the strands briefly. And then it's structured in such a way that after that little stretch of RNA-DNA helix is made, the new growing strand of RNA, coming out here in red, exits from the polymerase in a separate way so that the RNA doesn't end up tangled up with the DNA that it's being transcribed from. And we're going to see in a little more detail how it accomplishes all that. Well, if only 1% of the human genome is transcribed, how do cells pick which parts to transcribe? And transcription in general begins at regions that we call promoters. And promoters are just defined as regions at which transcription begins. But there are, in fact, sequence similarities amongst promoters that are organized somewhat differently between prokaryotes and eukaryotes. And this is one of the areas where prokaryotes and eukaryotes have rather different ways of handling things. If you look at the sequences in a prokaryotic genome, and again, in a prokaryotic genome, most of the DNA is used for something. In front of any given gene, you're likely to find sequences like this. There's what's called sometimes a TATA box, TAAT, at 10 bases upstream of where the actual start of synthesis is, and then TTG, ACA, of 35 bases upstream of where transcription is going to begin. And the enzyme in E. coli that synthesizes more RNA, or synthesizes RNA, is a complex. It's an alpha-2 beta prime multimer with a sigma subunit. And the sigma is, you can think of it as specificity. Sigma is the Greek letter S. You can think of it as a specificity subunit because the sigma subunit is going to affect which promoters that particular molecule of RNA polymerase can actually act on. And if you want to see that schematically, here is the +1 site, meaning this nucleotide will be incorporated into the RNA. Here's the -10 region, the -35 region. So it's a fairly simple set of clues that the RNA polymerase has as to where to begin. In eukaryotes, it's a bit more complicated. No, it's a lot more complicated. Eukaryotic promoters have something like a Tata box in the sense that they have a TA sequence typically, a little further upstream, around 24, so it's about halfway between the two eukaryotic sites. But then there is a huge variety of other upstream sequences that affect whether or not a promoter is used. So for example, a gene that might be used in 4 different tissues might have 4 different regions upstream of it that affect whether it's expressed in the liver or the muscle or the brain and so on. And in some cases, these regulatory units can be tens of thousands of bases upstream of where transcription starts. So the organization, the ways in which the regulation is organized for transcription in eukaryotes can be quite various. Another sign that it's done a different way is that the eukaryotic polymerase does not have a sigma factor. So in the prokaryotes, the sigma factor is sort of part of the polymerase, and it goes along with it. And a relatively small variety of sigma factors can accommodate the variety of promoters in the prokaryote. In eukaryotes, there is a really wide variety of transcription factors involved, and these are involved in both protein-protein interactions between the polymerase and other transcription factors or inhibitors or stimulators, and also protein-DNA contacts. So not just this area, but for perhaps quite a large bit of the landscape upstream of it, there'll be interaction with transcription factors. Now you can imagine that trying to identify the promoter, the region of DNA that's not transcribed but does turn on transcription, could be a little hard. If the genes are only 1% of the human genome, the promoters are a smaller fraction again because they're probably 1% or less of the gene area. On top of that, promoters in a cellular context are going to be hidden. They're going to be underneath histone proteins that are wrapping up all the DNA. There's going to be chromatin proteins that's going to vary between euchromatin and heterochromatin. The chromatin proteins can affect also whether or not a promoter is used. So for instance, in heterochromatin, the DNA is very tightly condensed. And most of the genes in heterochromatin are not transcribed. Whereas in euchromatin, the overall architecture is more open, more accessible. The histones in the euchromatin areas tend to be acetylated, and so more of those promoters and a larger fraction of those genes are actively being used. It's even thought that something like a histone code, you may remember we talked about that in DNA structure, may help select which factors are going to actually bind and be effective. Here's an example of acetyl-lysine. So up to this point, this is just ordinary lysine. Here's acetyl-lysine with the acetyl group from acetyl-CoA that's been added onto it. I had a thought back there in the heterochromatin that I wanted to say. Well, it'll come to me, I'm sure. Okay. So when we talk about trying to get transcription started in eukaryotes or people like us, there is a certain subset of things that are considered general transcription factors, that these are things that almost all promoters need to have in order to have transcription happening at that site. And there are several polymerases. We're going to start by talking about polymerase II. Polymerase II is going to be responsible for transcribing messenger RNA molecules. And as you might guess, it has perhaps the most complicated regulatory scheme because there's the greatest variety in outputs, that the tissues differ so much in which RNAs are needed. So transcription factors are usually abbreviated TF, and polymerase II has transcription factors that are called TF2 because they're transcription factors for polymerase II. So polymerase II needs TF2A, B, D, E, F, and H. And TF2D is perhaps one to pay a little more close attention to because it's quite a complicated and rich transcription factor. It includes a TATA binding protein. So you remember I said that there's a TATA sequence about 25 bases up from the start site. When you see a sequence in the DNA that's associated with something, it should trigger the suspicion, at least, that there may be a protein to interact with it. And this is the TATA binding protein. But TF2D has other things with it called TAFs, for TATA binding protein associated factors. And TATA binding protein does something kind of cute. It actually sits on top of the DNA and bends it, kind of like a heavy junior high kid sitting on a seesaw that's meant for kindergartners. Sort of sits and straddles on it. In addition to that binding at the TATA site, the TAFs that also are found in TF2D can be quite various, and those are the ones that are going to help confer specificity so that this gene is transcribed in this tissue and not in that one. And the TAFs, the TATA binding associated factors, many of them have parts of the protein that can interact specifically with sequences of the DNA. You may remember that I talked about how the major and minor groove had exposed potential hydrogen binding sites, and they provide some of the sites of interaction for things that are sequence specific. So these TAFs can sort of read that groove structure and say, that's my Velcro. That's the one I'll Velcro onto. One particularly striking TAF is TAF 250. It's called TAF 250 because it's a 250 kilodalton protein, so it's quite a good-sized protein, quite a big one. I like to think of it as the Swiss Army knife protein because this particular transcription factor has a lot of different enzyme activities. It has a kinase activity, so it has the potential to either phosphorylate the polymerase itself or to phosphorylate other transcription factors. It has a histone acetyltransferase activity, so it can modify the histones at the site where it's being recruited, which could increase the likelihood that that gene will be used again in the future. And it can let's see if we can say this. Ubiquitinolate. There we go. Ubiquitinolate histone 1. And when you ubiquitinolate something, you covalently attach ubiquitin to it. And by doing that, the ubiquitin targets that protein for proteolysis. So after you ubiquitinolate the H1, you are going to target that H1 to be degraded and taken off of the chromatin at that spot, and that helps open up the overall structure of the chromatin there and makes it easier for these other things to get in and do things. It also has, there are a number of these transcription factors that have areas that are called bromodomains, named after the first protein that had them. And what's interesting about the bromodomain is that it has a place on it where an acetyllysine can bind in a little pocket. So you can see these would be very good for interacting with areas of the DNA that had acetyllysine on their histone proteins. So after the TF2D has bound, other factors are going to come in and bind in addition. So you can see here, here's the TATA binding protein, and it's really kinked the DNA substantially. Here's TF2A and 2B have come in. Transcription factor 2H is easy to remember. TF2H has a helicase activity, so that's easy enough. You can see why TF2H would be needed. A helicase activity opens up that DNA double helix, allows access to the nucleotides, to the bases, so that you can make the complementary thing. And then after all of this stuff has gotten kind of plastered onto the promoter, polymerase II can dock with that complex of proteins. And we call that area where the DNA has been opened up the transcription bubble. We can get further regulation of these promoters from factors that act, in effect, act at a distance. I mean, it's kind of easy to see how something that's at 25 nucleotides from the start site could influence whether or not that base is going to be transcription or not, because you're right next to it, in effect. But there are other things that work at a much greater distance, things that are called enhancers and silencers. And enhancers and silencers are regions of DNA that can be effective on a promoter from considerable distance. In many cases, they can affect more than one gene downstream of them, for instance. So you might have an enhancer, several genes that are being enhanced, their transcription activity is going up coordinately, a silencer, and several genes next to it that are being silenced. Some of the current models for how enhancers and silencers might work is that the possibility is there for there to be looping of DNA so that you generate a higher order of architecture that brings together factors that wouldn't be otherwise. And they're showing you just a sort of a schematic here of a possible thing. Here's an enhancer region a long way away from a promoter, that if this stretch of DNA has an activator bound at the enhancer and general factors at the promoter, after the looping, you can see that this larger complex has now been assembled through a mediator, and that whole complex is more effective at initiating transcription than the other one would be by itself. This gives us much larger range over which transcription can be modified. There's a 10 to the 9th fold range over which you can have the lowest to the greatest levels of expression. So you can have really great levels of precision. And then each of these factors may have other factors that can modify their activity up or down, sort of like big fleas have little fleas on their backs to bite them, and so on and so forth, ad infinitum. Factors have factors. I've mentioned already that one enhancer can control several different genes. Other ways that genes can be controlled are through hormones. And hormones can affect gene expression in a couple of rather different ways. There are some hormones that penetrate the cell membrane. And if we think about the structures of these molecules, it may be more clear why that would be true. So, for example, steroid hormones or retinoic acid, which is derived from vitamin A, that branched lipid that we were looking at earlier, thyroxine, these are hormones that can go through the membrane. And inside the cytoplasm, they bind receptors that can recognize that specific hormone. So these are not working through the plasma membrane receptors we talked about earlier. These are using cytoplasmic receptors. For some of them, it appears that the receptor is in the cytoplasm when it's empty, and that when it is occupied by the hormone, it will then move into the nucleus. There may be some others that wait in the nucleus lurking, but certainly there are some for which it's documented that binding the hormone moves it into the nucleus. Then once that receptor hormone complex is in the nucleus, though that complex together can bind at promoters as part of the complex. So that's one way that hormones can regulate things. A second way that hormones can have their effect is more similar to what we were thinking about when we thought about the membrane receptors and the signaling cascade. Some of those hormones have what are called STAT proteins, signal, transduction, and activator of transcription. So these are proteins that are nuclear proteins that, because of the mediation of the cascade, so we don't know whether that's cyclic AMP or PIP3 or something like that, but some part of that cascade activates the STAT proteins, and then they dimerize, and in dimerizing they now are better at binding DNA, and they can bind to DNA and even bend the DNA in the process. If you think about it, it's a good idea to be able to coordinate gene expression, that it's going to often be the case that when you need gene A, you need 3 or 4 others that are related to that same function, that there's sort of a constellation of functionalities that need to be accommodated all at once. And so one of the functions of regulating transcription is to coordinate the expression of more than one gene. And this is done in rather different ways in prokaryotes than in eukaryotes. So if we look in prokaryotes, many of the genes in prokaryotes are in what are called operons. It's named by a Frenchman. I didn't pick it. And an operon is a string of genes, head to tail, that are for related functions. And we're going to make one messenger RNA with all of these genes in it. So the promoter here, here's the P for the promoter, is going to initiate transcription here, and then the polymerase will go clear through to the end and stop. And the messenger RNA that is made will have multiple places on it that a ribosome can come in and start making a protein. So this one messenger RNA is going to make 1, 2, 3, 4, 5, 6 proteins. This particular example is for something called the trip operon, and trip, of course, is tryptophan. And the tryptophan operon, as you're thinking of the structure of tryptophan, you can see that that's a complicated structure. You don't want to bother making tryptophan if you have tryptophan, right? It's a waste of energy to make tryptophan if you have tryptophan. And so the logical kind of regulation would be to only turn on the trip operon if you're low on tryptophan. And that's how it works. That when tryptophan concentrations are high, it's turned off. And when tryptophan concentrations are low, it gets turned on. And obviously, if it gets turned on and it's working and you accumulate more tryptophan than you need, it'll get turned off again. The way it works is there's one of these proteins in the trip operon is actually a repressor and made at low levels all the time. If tryptophan binds to that repressor, the tryptophan repressor complex will bind at the promoter and turn off transcription. So this is a repressible form of coordination. Slightly different logic. How does that project? Looks okay up there. How does it look on here? Okay. Looks okay on there too. Good. Slightly different logic on the lac operon. This is, again, a group of several genes. It has, in this case, 3 structural genes. One for an enzyme called beta-galactosidase, so it can cut beta-galactosides, things like lactose. Here's your lactose, which is a glucose and a galactose. The beta-galactosidase can split that bond, and now you have a galactose and a glucose. So that's what beta-galactosidase does. The permease allows lactose into the cell, which is a good thing to do if you want to use it. And the transacetylase we'll get to at the end of all this. But these are the 3 genes that seem to be involved with lactose metabolism. In this case, these are no use to you unless there is lactose present. You have no use for them in the absence of lactose. And so the logic of it is sort of the backwards of what it was for tryptophan. There's another structural gene for something called a repressor. The I gene makes a repressor protein. And this protein, the repressor, sits on the O site, the operator site. And that's part of what's called an operon. And it sits on the operator site, and it blocks the promoter. So this P here is the promoter that should stimulate transcription. And so the inducer will sit on the operator unless the inducer molecule is present. That's something called allolactose. This is the structure of allolactose. You can see that the bond is just a little different, right? And that molecule will bind to the repressor, it'll fall off, and you'll be ready to go. This last gene over here, the transacetylase, it may play a role in protecting the cell from non-hydrolyzable sugars and so protect it from some aspects of aging. The lac repressor is kind of a cute protein. I guess it kind of looks like Bozo the Clown. It's a dimer of dimers. So you can see here, I think, that this is a dimer. You've got your yellow and your red parts of it with this part of the DNA. And then you've got another dimer over here. And what it does is it grabs onto the DNA and holds it in a loop. And so here you can see the dimer of dimers in the context of the loop. So 1/2 of the dimer of dimers has this part. Here's the I site. Here's the structural genes down here. And what it does is it has this loop of DNA with polymerase one molecule of polymerase sitting there in the pocket, waiting. And what this means is that the response to lactose can be very, very rapid. That you just have to bind some inducer molecule to these, and they'll let go of the DNA, it'll snap open, and the polymerase will go roaring off. So it's a very good strategy for things where you want it off unless you have the thing you want. It's interesting to look at some common structural strategies for DNA binding proteins. These are DNA binding proteins that each of them have a role in controlling transcription. These are ones that are related to specific genes, turning on specific genes or turning off specific genes. And these motifs are motifs that turn up again and again in DNA binding proteins. One of them is called the helix turn helix, and it fits into the major groove, and you can see a little bit of a bend from it. The lambda repressor we just talked about has this helix turn helix structure to it. Another common motif is this leucine zipper. It's a coiled coil like this, and it might have either two molecules that are identical, or might have two different ones depending on what you're trying to regulate. And it again reaches around the sides here and sort of hugs the major groove from both sides. A third very common motif is the zinc finger. You may remember from when we were talking about protein structure that zinc fingers were one of the few motifs where the zinc actually played a structural role, and it also sits in the major groove. So all three of these families or motifs of structural proteins are taking advantage of what I was talking about in the major groove, that there are potential hydrogen bonding sites there that can interact in a specific way with different proteins. Okay, so let's see what actually does this. We talked a little bit about the RNA polymerase in prokaryotes. In eukaryotes, it's more complicated. First of all, there are several polymerases, and the ones in the nucleus have Roman numerals, so there's RNA polymerase 1, 2, and 3 in the nucleus. We mentioned that in RNA polymerase 2 does messenger RNAs. That's the one that's regulated by TATA binding protein and TF2D and that zoo of factors. But there also is a polymerase called polymerase 1, and it transcribes the ribosomal RNAs. Ribosomal RNAs are the bulk of the RNA in the cell. There are multiple copies generally for them. Maybe 80 to 85% of the RNA in the cell is ribosomal RNA because you need to make the machinery to make the proteins. And then polymerase 3, which transcribes transfer RNAs and a variety of other small RNAs. And we have a fair bit of transfer RNA, again, because you need that at translation. In addition to these three polymerases that are in the nucleus, we have polymerases in the organelles. You may remember I said that the chloroplast and mitochondria were unusual in that they had a double membrane and they had a genome of their own. They don't code for everything, but they code for some unique genes. And in order to do that, they have their own polymerases. So all of your mitochondria have an RNA polymerase in them. Some of the subunits for that polymerase are coded for in the nucleus, but they have their own polymerases. Polymerase 2, again, sort of follows the motif of a palm, hand, and fingers. Its job is, again, going to be matching up nucleotides and making a polymer. It requires a magnesium ions to do that. And it's going to synthesize the new chain from the 5' to the 3' direction again. All the polymerization of nucleotides works that way. Pol2 absolutely requires a template. There'll be no enzyme activity without a template. So it's similar to DNA polymerase in that respect. But it differs from DNA polymerase in that it does not need a primer. It simply needs a template of some kind to do it. One difference between RNA polymerase 2 and the primase that we talked about in DNA replication is that primase it doesn't really care very much about where it sits down, but it doesn't stay on for very long. It makes fairly short pieces of RNA, whereas Pol2 cares a lot about where it sits down, but once it gets going, it'll keep going for a long time. So during transcription, there's a brief period during which the newly made RNA, the new transcript, is in a DNA-RNA helix. Obviously, you're transcribing it. and the newly made base isn't going to be able to peel away instantaneously. And you may remember from our earlier things when we were talking about DNA structure that RNA-DNA hybrids look more like A DNA than they look like B DNA. So the DNA-RNA hybrid is rather similar to the overall structure of A DNA. The DNA molecule enters through two sort of jaws that help to pull it apart to make that transcription bubble. And at the polymerization site, the DNA has to kind of bend or turn to leave enough room for nucleotide to actually fit in. When that nucleotide comes in is a random event. There's no selectivity at the entry into the polymerase to select which one it is. I believe that's the structure of alpha-amanitin, but I'm not sure. I'm not quite sure. Oh, that's probably a good idea. Thank you for reminding me. Oops. If you would like to put your name on a card, would anybody like to help me distribute cards? You want to do it? Okay. If you would like to suggest study questions for the final, we can compile some study questions for the final. If you just want to put your name on the card, that's fine. Either way. So he'll distribute cards. When you're done, just make sure they're on the table at the end. It's a beautiful spring day. You ought to get some little grace on top of all the knowledge you're getting for actually turning up. So let me see. That's 20. 26. Right. Okay. All right. I think, if you don't mind, I think I will go on and talk about the RNA polymerase while I distribute cards. Is that all right with everybody? So this is showing you what I was talking about in a little more detail. The DNA helix entering, it has to be opened a bit to let the incoming nucleotide pair to it. It hits the wall to turn to the side. And there's actually a part of the polymerase that kind of turns down to hold the DNA in the space so that the whole process can keep on going. It's extremely important for RNA polymerase to be processive. You remember when we were talking about DNA, that the DNA polymerase could keep going for great long stretches on the leading strand. Well, in the case of RNA polymerase, if you don't make a whole molecule, everything's been wasted at that point. So RNA polymerase is by necessity and by design, well, by effect, extremely processive. And it will only read off of single-stranded DNA. And you think about the fact that genes can have anywhere from 1,000 to 1,000,000 nucleotides in them. You can see it needs to be very, very processive indeed. This is one that might be a little easier to see because we're not doing all the structural stuff. Here's the entering DNA. We've opened up a bubble. Nucleotides are entering here through the funnel. The magnesium there at the pores helping to neutralize the charge. The actual events of polymerization are happening in here. The RNA is going out the little back hole while the DNA reforms the helix off that way. And remember that this is a little stationary thing. We're feeding it through, right? There's the transcription complexes, so the DNA is being fed through this, and the RNA is exiting through that hole. Interestingly, it's been recently found that RNA polymerase can proofread. And you might wonder how RNA polymerase could proofread. And it turns out that if there's a mistake made, that error is going to distort the helix, as you might imagine. And distortions in the helix stimulate the growing RNA to back up. So here we have an elongating RNA polymerase. Some kind of a mistake happens, and so it transiently backs up for a little bit, cleaves off the bad bit of the RNA, but you still have the slightly older RNA that you recently made on the RNA polymerase in contact with the DNA. And so once the distortion has been cleaved off, you can pick up again and start going forward again to make a second try at it. And it turns out that TF2S, transcription factor 2S, is actually a nuclease. Either it has its own nuclease activity or it's stimulating the RNA polymerase to do that. We're not quite completely sure yet. Now, we've been talking about transcription factors. We've been talking about initiation, trying to find the place to start synthesis. And there's a lot of different factors involved in trying to start synthesis at the right spot. But most of the time that the RNA polymerase is doing something, it's elongating. It's just doing the same thing over and over and over again. And the elongation phase actually turns out to be a fairly significant part of RNA polymerization. When we look at an initiation complex where we've got the RNA polymerase here with this thing a little sticking up, and I'll tell you what that is in a second, and we've got general transcription factors and other modulators that are sitting there. What we see initially is that a lot of little short transcripts are made and then thrown away. Once in a while, the RNA polymerase will get it and keep going and do the whole full-length transcript. So why is it sometimes one and sometimes the other? It turns out that RNA polymerase has on it a C-terminal domain, an end of the molecule that's sort of long and floppy. And the C-terminal domain, or CTD, has this sequence, tyrosine, serine, something, proline, threonine, serine, something, proline, serine, has that peptide sequence 52 times. 52 times. And the serines that have the star next to it are ones that can be phosphorylated. And so in the absence of that phosphorylation, you may remember that one of the transcription factors was a kinase. There are other kinases as well. When those serines are phosphorylated, this C-terminal domain now has a whole lot of phosphates sticking on it, and that increase in phosphorylation releases the mediator. This CTD domain that's long and floppy and gets phosphorylated, sometimes been called the negative noodle, which is a way to remember it in a vivid way because all those phosphates are negatively charged. When an RNA polymerase has taken off like this, it has elongation factors that are now associated with it. It will be very processive and keep going down that gene. It'll leave behind the general transcription factors. And so that makes this promoter more likely to be picked for another round of transcription than one that has not had that experience. So this is one way in which expression at a particular site gets turned up. Let me see. Has everybody had a chance to think of a review question? Okay. Just trying to decide if we want to hit histones. Yeah, we can do the histones quickly. You might have been wondering right now, what about histones? So we talked about that beads on a string appearance, that you have DNA wrapped around the outside of the histones. It doesn't look terribly accessible. And this may be one of the reasons that histone acetyltransferases play a role, that in acetylating those lysines, the histone's charge is partly neutralized. It's not as tightly associated with the DNA. It makes the affinity of the DNA for the histones or the histones to the DNA lower, and that allows it to breathe and keep open. Interestingly, it does not make the DNA completely come off the histones. There seems to be a capacity for the RNA polymerase to sort of go around the histone cores without actually having to knock the histones out completely. And this is probably important because if the histones carry a code that's important for turning transcription up or down, you wouldn't want them to have to fall off every time in order to go through transcription. And there are, in addition to this, other elongation factors that can improve synthesis as well. So why don't we call that a week? Have a great weekend. Enjoy that beautiful weather out there. If you have cards for me, feel free to leave them down here.